I hope you guys can all join me in welcoming Joel McDade. So Joel is trained as a cell biologist and with a degree from Michigan in molecular physiology. Since that time, he's actually spent most of his time here in Massachusetts, and it's currently coming to us as the technical sales director from Quantum Sci. So let's please welcome Joel McDade.
Thank you. Do a little test here. Oh, I don't want to succumb to the technical issues. All right, that's okay. I think maybe, did he have it or, let me see. That does it. All right, cool. Yeah, so first of all, thank you. It's great to be a biologist here with all of you wonderful proteomics folks. It's very humbling, but I appreciate the time that you guys are sparing to listen to my talk. And I also recognize that I'm standing between you and the bar, so I'll try to keep this short. Yeah, so my name is Joel McDade. I'm the Director of Technical Sales with Quantum SI. I'm here to talk to you today about what we feel is an orthogonal technology for protein analysis. And I'll tell you a couple case studies. I'll introduce the technology for those of you that perhaps haven't heard of it before. And then I'll go into a few case studies that highlight the orthogonal value of this technology, one of which you might not necessarily think, but it's in the mRNA therapeutics space. And I'll talk a bit about that, and then I'll talk about a few papers that are probably closer to what you guys think about on a daily basis, which is how our technology couples with mass spectrometry. And then we'll hopefully have some time for Q&A. All right, so this is going to be obvious to everybody in this room. It's not going here. Hold on. But the proteome is complex, and this complexity brings a variety of opportunities, but also challenges, right? So on the first front, sort of proteins are complicated. There's a lot of proteoforms. In order to get through all of these proteoforms and understand their biology, you obviously have to sift through a lot of proteoforms that aren't represented at the genomic level. In the case of engineering proteins that you want for a particular function, you have a swath to choose from, but of course then you have to screen through all of these proteins through some manner, and that's cumbersome. And so, obviously, if you talk to synthetic biologists, which I'm from that background, you talk to folks about this design, build, test cycle. I'm going to design a thing, I'm going to build the thing, and then I'm going to test the thing. That's a pretty painful process, and so tools that accelerate that are of significant benefit to not only drug developers but also basic biologists. And at Quantum SI, we feel novel tools are needed to address these challenges, and we hope you directionally agree with that. And in order to really expand the toolkit that proteomics, protein analysis folks have, we launched what we call the Platinum Pro. So this is the second generation of a system that was launched 2 years ago. We position it as really an easy to use option for doing basically bottom up proteomics. So we could really teach someone new to protein work how to use the technology. So it's a great orthogonal technology. The basic foundation of how it works is shown here. So the scientists would generally start with their protein or proteins of interest. You generally enrich for those proteins, and then you digest them using a series of off-the-shelf kits that we have provided. You add a linker to the peptide library that's generated, and then you flow those peptides onto a chip. Okay. The individual peptides then sit down with their C-terminus through a biotin-strobivatin linkage at the bottom of the well, and so you have these individual peptides in an individual well. In Branford, Connecticut, we do directed evolution to come up with these what we call recognizers, which are essentially fluorescently labeled proteins that are designed to recognize a series of amino acids, and they're specific for that. And the signal that they produce when they bind is actually unique depending on the amino acid, and it's influenced by downstream amino acids. So we have a whole algorithm. I hate throwing AI into the mix. I think it's a little bit kitschy, but we generate a lot of data using these recognizers. We model off that data. The software knows what it's supposed to see, or what it's actually looking at when it sees it. And so the basic premise is you have this peptide library, you flow it onto a chip, individual wells get individual peptides, you get a series of binding events with these recognizers. You generally get tens, if not hundreds, of binding events per amino acid. So it's a very data-rich technology. So let's take our NGS hat off, right? We don't need that many reads with this technology to be confident. We'll get into more of that later. But each of these signals will vary depending on its, which amino acid it is. Here you can appreciate the intensity differences between the different amino acids. There's other differences I can get into, like how long these actually stay bound. We call that pulse duration, and also the decay curve, right? So all of these things are captured by the software. This particular thing is squished in time, so you don't get to appreciate all the kinetics, but they're there, trust me. And then this is actually taken and generated what we call a kinetic signature plot. All of this is automated, and then that kinetic signature plot is actually what's used by the software to go in and say, okay, let's look across the proteome. What does this peptide look like? Okay, and we do that all automated, right? So essentially, the workflow-wise, you set up a run virtually through your laptop, and then you load the system. The run takes about 10 hours. The data transfer is automated, and so is the analysis. So you run a primary analysis. This is really calling that kinetic signature plot, and then you run a secondary analysis, and that can be either, hey, I know what I'm looking for, please align against this protein, or hey, I have no idea what I'm looking for, please infer what that protein is against the proteome. So that's the basics in a couple minutes there. The data, what does the data look like? So we provide insights at really the protein level. So for protein level, peptide level, and amino acid level. So here we're showing PDL1 sequenced on our technology. At the time, we got about 8,000 reads that are attributed uniquely to PDL1. You see each of the individual peptides that have their own number of reads per peptide. You also get an FDR score, which is modeled off of what you folks generally would do. Again, not a bioinformatics person, I can't give you the exact details, but that's at least what I'm told. And then down all the way to the amino acid level, you actually get data on each amino acid that is recognized by the technology. And by data, I mean pulse durations, how long does that binder stay on, and you get a histogram of that. That can be impacted by things like PTMs. And so we're working through some of that biology now, but we're very excited about the value that this sort of analysis can provide. And some other stuff that I won't get into in the interest of time. But all of this is produced at a glance, completely automated, so you don't really have to do anything for that. All right, so that's the core technology. What are we doing with it? There's a variety of things that we're doing with it on the application front. The first story I'll tell you is about protein barcoding. And this is the idea that we can generate a highly sequenceable protein barcode that we know to be sequenceable and engineer it on proteins and perform things like pooled screens. And this is of value because sometimes those pooled screens are performed with the same exact protein or highly similar variants that are very difficult to resolve on traditional technologies. And the use case that I'll show you is mRNA. You can confirm proteins, basic QC, right? This is not necessarily earth-shattering, but to some, it beats a Western blot. You don't need an antibody for it. Things like protein-protein interactions. So we're not necessarily going to get the same amount of proteins that you guys get, full disclosure, on a mass spec run, right? But as a biologist, someone looking at protein-protein interactions, it's a pretty simple solution for that use case, and we're building out workflows for that. And then finally, we're getting really creative with it. So for instance, with our recognizers, you can actually take your favorite protein of interest, turn it into a recognizer with our chemistry, and perform binding assays on chip. So one of the things we did is we made a new binding assay using a BCL2 recognizer and some peptides that were thought to interact with BCL2 and potentially inhibit BCL2, and we're starting to perform binding assays there. You can do that on SPR, but the challenge is that with SPR you have to know what you're looking for. With this, you can perform binding and then sequence to get the identity. So that's an application that we're working on. I don't have data to share, but hopefully we will soon. And then we're applying this to, across cell and gene therapies, vaccines, antibody biologics, all the things that you'd probably expect. So in order to kind of talk you through the barcoding application, I need to give you a bit of a background on mRNA therapeutics if you don't already have it and some of the challenges that they face. So I assume all of you have heard of mRNA LNP-based therapeutics and all the things that we're very excited about as it relates to immunogenicity, ability to deliver outside the liver, all the different possible benefits of this technology. The scientists start with a large number of potential candidates. So you can vary the lipids, you can vary the buffers, you can vary the mRNA chemistry, you can vary the cap, 3' UTR, et cetera, et cetera, et cetera, right? And the goal is really to get down to, selective reveal here, a small number of candidates that are ideally suited, right? And ideally suited meaning they get to the tissue, they translate, they produce protein, right? And they have a variety of tools to do this. So they have cell-based assays, they have in vivo mouse studies, they have in vivo non-human primate studies, right? Pretty obvious. The problem is that for these particular candidates, generally speaking, every single one of these codes for the exact same protein. So although optimizing mRNA delivery and translation is essentially the essential assay for knowing which one of these candidates wins, you can't multiplex them, right? If you do multiplex them, you're generally looking at using NGS, which paper after paper coming out showing that, hey, look, delivery doesn't equal translation. You can have a lot of mRNA there, and if it has a lot of ribosome on it, actually, hey, it gets degraded, so the total protein output's lower. So that's part of the problem with this is now you're left with this costly arrayed screen where every one of these guys is obviously there's ethical concerns, but they're like 25 grand a piece, right? So this is not a friendly situation to be in, although you need to do it, right? So where do we come into the picture? So we had this technology that we developed in our lab, which essentially what you do is you append to your gene of interest, your mRNA of interest, a highly sequenceable protein barcode. So it's your gene of interest encoded as RNA, an affinity tag, a linker, a lyse cleavage site, and then a highly sequenceable barcode. Okay, we've gone through in this publication and talked about design and complexity and limited detection and all these sorts of things that I won't bore you with it today. But essentially the point is that we can detect these barcodes and sequence these barcodes, and each one of these barcodes is going to be unique for a given candidate. So you can pool all of these candidates together and perform an in vivo or an in vitro pooled screen with a direct protein level readout. And this is very hard to do with traditional technologies, right? And so we've published this paper. It's in the process of being formally published. It's in bioRxiv, where essentially we describe the workflow where you would take your protein of interest. One can envision other applications for this: antibody biodistribution, uptake, secretion, variety of things. But in this case, we've just defined the technology, and we have all the different components, broken out essentially. And the workflow takes about 3 hours from the time you have a sample to the time you're sequencing. And so it's very, very fast to get this data, and it's quantifiable because the output is reads, right? So it's a very nifty technology to be able to apply to some of these questions. And so we've published the actual biology work in a technical note that I'd encourage you guys to check out if you're interested. But it describes essentially the process of putting these mRNAs into an LNP, the variety of LNP candidates, mixing those together, injecting them to an animal. The barcoded mRNA, the winners, so to speak, get translated. Transcribed shouldn't be there. Sorry about that. You then extract the tissues, and then you purify out the barcodes and sequence them on platinum, and essentially what you get is a rank order of your candidates, and so you're able to uniquely attribute whatever those conditions are with the barcode, and so you get a pretty elegant solution. And obviously the costs on this, if you drop the number of monkeys you need by a factor of 8, you're saving a lot of loot, so it's pretty exciting. Okay, take a step back. Now we're talking about just protein characterization. So we published a paper recently in bioRxiv. This is the Neil Kelleher paper, where we're looking at the complementarity of our technology with something like top-down proteomics. And so we're looking at this case at IL-6, and the idea is that we're taking a top-down approach as well as the bottom-up approach with single molecule sequencing. And so what we saw is pretty neat. We generally picked up hydrophobic peptides on our technology, and versus some of the hydrophilic peptides tended to show up on top down. Overall, the coverage was complementary. It was complementary, so we got about 52% coverage total. So again, it's just speaking to this idea that that we could be complementary with this technology. And then the final case study I'll share is with Gloria Shankman at UVM, University of Virginia, and she's interested in tropomyosin, which is actually a gene I studied, ironically. But this has a lot of heterogeneity between these tropomyosin isoforms, but they're highly similar. And so some of them will actually cause disease, even though they're just point mutations. And some of the point mutations are things like L to I substitutions, which are not necessarily ideal for mass spectrometry based, though you guys, I'm sure, will argue with me about that. But similar masses equal similar readout, and we're able to discern some of those on platinum because we don't care about mass necessarily, right? And so we're also starting to look at things like phosphorylation on the technology that's shown in the paper. And so I'd highly recommend you guys take a look at that as well. This is for those of you that have been following us. This is kind of like just a snapshot of where we've been and where we're going. So, we launched when we published this paper in Science back in 2022, sort of came to market in December 2022. I joined right around, I think it was like somewhere in 2023. And then we very quickly since then have launched, we've doubled the number of recognizers. We've quadrupled the amount of occupancy on the chip. We've launched several new kits improving the chemistry. We've launched a new system. We've launched new software. The technology is really, really, really moving quickly. I haven't been at a company where the technology moves as quickly as this does. And so we're very excited about where we are today, but of course where we're going. And we would love to have you guys on board as if you ever want to test the system, anything like that, just let me know. With that, I'll take any questions, and hopefully I'm under time as promised.
Let's thank Joel one more time.
Thank you guys.